Thank you, everybody, for joining. This is our last Biotech Connector Speaker Series of 2023. This year went way too fast. This is I'll start from the beginning. So thank you all for joining, for the people that are here in person, and we also have a great crowd online. I am Dr. Maggie Scully, the Partnership Development Office Director at the Frederick National Lab for Cancer Research. The Biotech Connector event is a collaboration between the Frederick National Lab and our great partners at the Frederick County Chamber of Commerce. The event is a quarterly series that bridges our local biotech community to get an inside look of some of the things that we're all doing. A chance to share your expertise, as well as an opportunity to network around coffee and pastries. So switch to the next slide. FNL is a collaborative enterprise, and we are always interested in partnering opportunities. So here's a list of some of the contacts of people if you're interested in partnering. Also, for anyone interested in subcontracting and acquisitions, contact information is there. And for anyone looking for new career opportunities, there are certainly a good number of career opportunities on our website, and you have the contact information there as well. We'll also put this contact information in the chat box for the people online. Okay, so today we have an amazing event focused on innovation in mass spectrometry. So I'll go quickly through the logistics. I'll intro everybody now. We'll move through the speakers one by one. There'll be time for a question or two after each presenter. When prompted, please stand up and state your question. There are microphones in the room, so they can hear you. We don't need to pass around a microphone. For the people online, feel free to unmute yourself, or we can have someone take that question from the chat box if you feel more comfortable that way. Okay. Now I have the pleasure of introducing our speakers for today's event. First, we have Dr. Caroline DeHart in the RAS Initiative Mass Spectrometry group. She's the leader of the Mass Spectrometry group in the Cancer Research Technology Program here at the Frederick National Lab for Cancer Research. Caroline's team focuses on employing cutting-edge protein analysis methods to better understand and target canonical RAS isoforms, RAS-related proteins, and RAS-dependent signaling pathways. Dr. DeHart will be presenting the ongoing efforts to improve the immunoprecipitation top-down mass spectrometry characterization of RAS proteoforms within malignant cell lines, with the ultimate goal of better understanding and inhibiting RAS-dependent oncogenesis. Next, we'll have Dr. Robert N. Cole, who's an assistant professor in the Department of Biological Chemistry at Johns Hopkins University. Dr. Cole also directs the Johns Hopkins School of Medicine's Mass Spectrometry and Proteomics facility, which he established in 2000 and provides proteomics services to hundreds of investigators, both at and outside of Johns Hopkins. Dr. Cole's research is highly collaborative and focuses on developing, optimizing, and applying innovative mass spectrometry techniques to identify the functional characteristics of proteins in health and disease. His team is currently developing proteomics strategies to identify albumin adducts in plasma to monitor exposure to environmental carcinogens and assess health risk. Our final speaker, and I'm sorry if I don't get this right, Dr. Ventzi Hristova is an associate director in translational medicine at Amgen. Dr. Hristova just recently transitioned into a new role at Amgen from AstraZeneca, where she established a clinical omics team focused on mass spectrometry proteomics and supporting all stages of drug development. Dr. Hristova is a clinical proteomics expert and a team manager leading translational studies aimed at patient proteome profiling to support precision medicine efforts through biomarker and therapeutic target discovery across disease areas. Okay. Now it's my pleasure to welcome our first speaker, Dr. Caroline DeHart. Thank you, Caroline.
Okay. We're going to see if we can get a laser pointer up here. All right, morning. Can everyone hear me? All right, thank you so much for coming out today, and also thank you to the organizers for the opportunity to present our work in this forum. For those of you who are wondering, a proteoform is a distinct molecular form of a protein. So any given proteoform incorporates all relevant information about variations in primary sequence, as well as modifications such as SNPs and post-translational modifications. We analyze proteoforms by top-down mass spectrometry, where we send the intact denatured protein molecules directly into the mass spectrometer for a two-part experiment. We first generate an MS1 or intact mass spectrum that allows us to get an accurate monoisotopic mass. We then bombard the protein molecule with nitrogen, helium, or free radical ions to generate protein fragments, or MS2, which we can then determine the fragment ion masses and identify the protein, identify post-translational modifications, and determine where these modifications are located. This method of mass spectrometry is in contrast to more traditional methods, which are peptide-based or bottom-up or shotgun proteomics, what have you, which entail digesting the protein molecules with an enzyme to generate proteolytic fragments or peptides, which can then be sent into the mass spectrometer for the same type of analysis. The intact or MS1 spectrum of each of the peptides, the MS2 fragmentation to identify the proteins within the sample, the modifications, and allow specialized applications such as targeted quantitation. At the RAS Initiative, we're very interested in RAS proteoforms because the RAS genes are among the most frequently mutated in human cancer, with different isoforms representing a high percentage of mutations in distinct cancer types. There's also a large variety of mutations across cancers located within different tissues. These proteins have represented a formidable challenge to design successful targeted inhibitors to slow or eliminate RAS-dependent oncogenesis. So our goal is to learn as much as possible about the proteoforms resulting from each mutation in each of these cancer types, so we can then design better targeted inhibitors. However, the RAS proteins are incredibly challenging to analyze by standard proteomic methods. They're expressed at low endogenous abundance. They have very challenging and labile post-translational modifications, but the most significant challenge is the high sequence identity. These four proteins, HRAS, NRAS, KRAS4A, and KRAS4B, are nearly identical, save for the C terminus of the protein, where the key post-translational modifications are located. However, the most frequently mutated codons, 12, 13, and 61, are located towards the N terminus of the protein. So if you digested these proteins with an enzyme into peptides, particularly from a complex biological sample where you had all four present, it would be very challenging to figure out which mutant resulted in which change in post-translational modifications, or to link any modification or mutation to a given isoform. However, by top-down mass spectrometry, despite the high similarity between the proteins, they are distinct enough that we can clearly distinguish between them by intact mass. We have the recombinant examples across the top and the endogenous examples across the bottom. We can clearly tell which proteoform is which. We can also directly link post-translational modifications to a given proteoform. So we can say, for example, this acetylation is present on NRAS and only NRAS. Moreover, we can clearly distinguish the wild type and mutant forms of the protein. So this example of KRAS4B shown here, you have two sets of proteoforms that are separated by the mass of the G13D mutation. So our workflow combines a strategy of RAS immunoprecipitation followed by targeted top-down mass spectrometry that has been described in detail in a series of publications, the most recent of which is available here. While this workflow was originally designed for a QEHF mass spectrometer, we have adapted it to the Orbitrap Fusion Lumos mass spectrometer that we have here at the RAS Initiative. We combine reverse phase liquid chromatography with high-resolution accurate mass spectrometry using Fourier transform, or FT, mass spectrometry and an Orbitrap mass analyzer. The most important features of the method to take away are that we first do a screen to identify which proteoforms are present in the sample, and then do tailored on-the-fly targeted methods for each proteoform population. And the reason we need to do that is shown in these two samples that were prepared side by side and run back to back. You can see that the population of targets is markedly different between these two contexts. So if we tried to run a one-size-fits-all method, we would miss a lot of important information from within these samples. We also fabricate our own nanocapillary columns, and we use a home-built nanoelectrospray ionization source, as we have determined that the conditions provided by this setup best retain the highly labile post-translational modifications of interest on endogenous RAS proteins. And to walk through what the data look like and what you'll be seeing for the rest of the talk, this is an example from recombinant KRAS4B of an intact mass MS1 protein spectrum showing the charge state peaks generated by electrospray ionization. We can also zoom in on a given peak to get better detail on the mass shifts or adducts such as oxidation, which are marked with an asterisk here. And then for MS2, each of these flags, which are colored by convention based on the type of MS2 fragmentation, represents a fragment mass that has been successfully matched to the proteoform sequence using a software called ProCyte Lite. The quality of the match is shown by this probability-based score here. And then there's also post-translational modifications that are also colored by convention, indicated by the color-coded residues here. This started as a multi-year collaboration between the FNL, the NCI, the NRTDP at Northwestern, and the National High Magnetic Field Laboratory with the goal of examining KRAS4B proteoforms in colorectal cell lines and tumors. We first adapted the method for the QEHF mass spectrometer using cell lines, published this initial study, reran the samples from this initial study on the 21 Tesla FT, so it's Fourier transform ion cyclotron resonance mass spectrometer at the National High Magnetic Field Laboratory, which is a different method than the Orbitrap mass analyzer, entailing this minivan-sized magnet here, which can generate nearly atomic resolution of protein molecules. This is the current world record holding mass spectrometer of this type. And it really illustrated just how much we had missed in our first run. So after taking that knowledge into account and applying it to an expanded panel of cell lines in patient tumors, we found a wealth of novel proteoforms, including one lacking cysteine 185 and all associated post-translational modifications. However, for each proteoform that we identified, dozens remained uncharacterized, including NRAS, certain mutations that we analyzed, and a number of post-translational modifications we expected to be present. And these data from the first run on the Lumos using the published optimized parameters shows the problem. We have a lack of sequence coverage on the endogenous proteoforms from our MS2 fragmentation method. We're ejecting a number of key post-translational modifications due to the force of the bombardment, and we're losing a lot of key detail that would help us both confirm the presence of the mutated residue and identify post-translational modifications. So what we've done, we first started with recombinant versions of each of the four isoforms, KRAS4A, KRAS4B, HRAS, and NRAS, and we developed targeted MS2 fragmentation methods with isoform-specific parameters, focusing on getting the best sequence coverage in the shortest possible time, as that's imperative for successful characterization of their endogenous counterparts. We also adapted some of this optimized method development into better understanding the relationship of KRAS4B in a public library of KRAS4B targeting compounds, bringing new insight that standard pharmaceutical methods are unable to deliver, which will be featured in an upcoming publication. We also analyzed our favorite model system, which is a flag-tagged RAS construct in a panel of cell lines. This can be expressed at high concentrations, purified to near homogeneity, and it bears endogenous processing and post-translational modifications, making it an ideal system for method development. So going through a panel of mutants and a variety of cell line backgrounds, we were not only able to determine the MS2 parameters that would retain the most highly labile post-translational modifications, for example, geranylation of KRAS4B, but also answer some fundamental questions. If you study different mutants of KRAS4B, would you find different proteoforms? And as you can see, yes, the KRAS4B G12C has a large number of proteoforms that are distinct from those present in the G12D and wild type, as our eventual goal is to link these proteoforms to downstream KRAS4B function in normal and oncogenic cell contexts. We also performed a considerable amount of method optimization to retain palmitoylation, shown here for HRAS and NRAS. This is a formidably challenging post-translational modification to analyze by proteomic methods. However, we are now able to retain the modification and directly visualize and quantify its presence, showing that it is present on the expected residues in the expected numbers for both the flag-tagged and endogenous constructs purified from malignant cell lines. So to highlight why we're working so hard on this optimization over the past couple of years, we have identical samples analyzed by our original method on the left and our newly optimized method on the right. So we've managed to obtain a dramatic improvement in proteoform signal from these malignant cell lines, thus enabling us to visualize for the first time novel proteoforms. So for this example of KRAS4B G12C, by targeting this proteoform, we were able to localize the glutathione to C12. We were also able to determine that about equal proportions of the G12C proteoform had the glutathione or had an empty C12 residue. We were also able to find similar results in this homozygous G12C mutant cell line, although we have seen this in every KRAS4B G12C cell line we've analyzed, again localizing by targeted MS to the glutathionylation to the C12. We were also able to identify additional unexpected G12C proteoforms, including a disulfide bond towards the N terminus of the protein. You can see where the sequence coverage drops out due to the altered structure of the protein in the gas phase, as well as the presence of a sulfinic acid or an oxidation at cysteine 12. And again, we've seen this in all of the cell lines that we've analyzed. We've also, due to now being able to retain the palmitoylation, started to explore NRAS proteoforms, which we were previously unable to characterize using our optimized methods. So in addition to the canonical palmitoylated proteoform here, we found some interesting variations. So on the left, we have a proteoform that's palmitoylated but not farnesylated. And on the right, we have a proteoform that's palmitoylated and phosphorylated. We also have this doublet of acetylated proteoforms here, where the acetylation can be present on lysine 147 or lysine 160. There is equal MS2 support for both. We've been analyzing a panel of cell lines, as I've mentioned, and we've seen repeats of these proteoforms. So we're trying to start to elucidate what the significance of these modifications and their difference in relative abundance might be in terms of the tissue and mutation backgrounds. But we still have a long way to go and a lot of work to do. So to tackle things that we might be missing, we analyzed a panel of these flag tag RAS constructs from a PANC1 cell line background and cataloged all of the abundant proteoforms that we saw for each of the 4 isoforms, which led to a number of surprising revelations, most prominently that a number of the post-translational modifications described in the previous literature had been ascribed to the incorrect isoform. We're still following up on which of these might be present in cell lines and eventually tumor backgrounds. And then Dr. Grace Scheidemantel, who joined us in May, has been working on our new system, our Orbitrap Exploris 480, to develop novel targeting methods so we can get higher sensitivity and higher speed in detecting these proteoforms for a variety of future applications, both proteoform biology and inhibitor development. But in the meantime, I'd like to thank all of the people who helped make this happen, as well as the funding and my amazing group, and you for your attention. I'm happy to take questions. Hi, Tommy.
So I think you alluded to it, but are you planning on looking in tumor samples from patients?
We do. We need to fix the sensitivity problem. So the Exploris is 20-fold more sensitive than the Lumos in terms of proteoform detection. So we need to get the MS2 fragmentation up to the caliber of the Lumos and then start tackling the tumor proteoforms because they are not nearly as abundant as you can get from a billion cell pellet. Hi, Anna.
So Caroline, on one of the last slides, you had a G mutant that was also beta-carolinized, right?
Just G12C for now.
Okay. So it just sits on the mutation site, and C12 mutation site, and you haven't seen any modification to C180. Should we solve them both?
We have not seen C118. We have seen modifications on other cysteine residues that are supposed to be, for example, palmitoylated.
Oh.
All right. Thank you so much.
Okay. Hi, I'm Bob Cole. I'm from Johns Hopkins University, and I thank the organizers for inviting me out to present some of the work that's ongoing in our lab. You may have heard it before, but what if I said if I could take a pinprick of your blood and tell you what environmental toxins you've been exposed to in the last 3 months, would you want to know? That's great, because I'm going to show you our first steps into actually trying to assess personal exposure to environmental toxins by a mass spec-based method, which we call pan-protein adaptomics. Okay, your risk of developing disease is a component of your genetic background and your environment. And it turns out the environment's a lot more important. And that was supported by this work in 2000 by a group in the Netherlands that looked at 11,000 incidents of cancer in about 10,000 pairs of twins, both fraternal and identical, and found that genetic component factors may account for up to a little less than half of the incidents, but clearly over 50% of the contribution to developing the cancer was attributed to environmental factors. So environment has a big play in your risk of developing cancer. And we're assaulted by environmental toxins all the time, most notably by air pollution, either by our activity, by accidents, and by burning stuff that we shouldn't be burning. We're also assaulted by environmental actions, such as the wildfires in Canada recently. This is the level of smoke in and around Baltimore and Maryland this June during the Canadian wildfires. And if you look at the satellite imagery of this, you can see that air pollution knows no geopolitical background boundaries. So we're all exposed to these kinds of things, no matter what our economical, social status is. Okay, so what's in air pollution? Air pollution is a complex mixture of many different compounds. Most of them are generated by burning fossil fuels. All of them by the CDC is classified them as known or probable carcinogens. And the International Agency for Research in Cancer, or IARC, has classified air pollution as a class 1 carcinogen. So how do we go about measuring our exposure to these toxins? Well, you can use real-time air pollution monitor stations that are located all over the world. There's a website, the World Air Quality Index website that you can find any place that you want to see the up to the minute or level of air pollution at that station. And as you can see, there are stations that show good air quality, the green, and as you go towards brown or purple, less good air quality. And the problem is, if you're sort of in between stations like Johns Hopkins, what is my exposure, real exposure at that time? Another way to do this that gets a little closer to your exposure is these wearable pollution monitors that you can wear in your clothes or wrist, and they all have these neat apps that allow you to see the particulate matter levels and the levels of volatile organic compounds to get an idea of what the air quality is where you are right then, and you can decide whether you want to go for that run or go back inside. But this doesn't really give us the actual internal dose, because that's where the real risk, assessing risk for disease development is. And when we incorporate toxins into our body, they or their metabolites will modify proteins and affect the functions of those proteins. So another way of going and seeing, determining the amount of internal dose is look at what is decorating your proteins. And a nice target is albumin. And the reason why albumin is a great target is that there is this cysteine at position 34, and that is on the surface of the protein, and at the pH of blood, it is deprotonated and can accept electrophiles. It also is, albumin is quite accessible. You just take a blood sample. It's abundant. It's about half the total protein in your body, in your plasma, and it has a long half-life, so you can look at accumulation of adducts over time. And the Rappaport group originally showed that the cysteine 34, a lot of different things can bind to there. So it is one of the hot spots in this protein. But there's a lot of other sites here that can potentially pick up and bind adducts. And so what Josh Smith, who is a research associate in my collaborator John Groupman's lab, and is probably going to be looking for a job soon, and Bob O'Mealy and Lauren Devine in my lab developed this nice workflow in which they take 2 microliters of plasma, they digest it with trypsin and lyc using pressure cycling digestion, and then that gives you peptides, which then can be analyzed by high resolution mass spectrometry and nanoflow tandem mass spectrometry for high sensitivity. So the opposite of what the previous speaker was doing. We're going bottom up, she was going top down. It's a little more complex than that, and this is what I call our chutes and ladders workflow. But I want to point to this area right here. After we get through the sample prep and the mass spec analysis, this is a good output point where we are seeing mass shifts in the peptides that give us an idea of what the adduct might be, and then the mass shifts on the fragment ions, which give us a position of that adduct on an amino acid within the protein. So this gives us lets us know the mass shifts and then the purported adducts, the sites of these adducts in the albumin, and by looking at the number of PSMs, we can get a sort of a qualitative idea of the amount of protein, amount of the adduct and in the protein. And this is important because the next step of the data analysis is currently very labor intensive and lots of data. And with lots of sites, you want to be able to right now pick and choose what you're going to be looking at, because at this next step we quantify the amount of the peptide, the adducted peptide relative to the heavy standard of the unadducted peptide, and do some normalization, put it against standard curves, and eventually come out with the output of relative abundance of that adduct per milligram of albumin. When we look at albumin, we're seeing in this way, we see peptides throughout the protein, as shown by the sequences in yellow, and the green highlighted amino acids are all the amino acids that we've seen an actual adduct on. So we're not restricting now to just looking at what's happening in the N-terminus of this protein, it's cysteine 34, but we're seeing adducts throughout the protein. This is important because it gives us more of information of what is being exposed to. When we looked at the study in which they were following non-smoking women in China over 84 days and took 3 time points of these 84 days, we're identifying 68 adducts on 21 different sites in albumin. Clearly, cysteine is the most popular place for an adduct to go, but that's only half the story. It's less than 50%. There's another 50% is at other positions within the protein. And so there's a wealth of information to try to develop either a signature or other biomarkers for exposure. This just shows that of some of the cysteine 34 adducts that we can measure, and we can see that some of them will change over time. But again, we're not just looking at cysteine 34, we're looking at non-cysteine 34 adducts, methionines, lysines, tyrosines, histines, and some of these also changing with time and exposure to increasing air pollution. We're still trying to figure out what this all kind of means and how it plays out in terms of exposure. But if I draw your attention to this trioxidation, which is increasing with increasing exposure, and oxidation, at least one of these methionines, is increasing exposure, it appears that this might be a sensor of oxidative stress in exposure to increasing air pollution. And oxidative stress is not good for health or your immune system. And also, we know that there's a report that oxidized cysteine it affects the 2-year survival rate of individuals with cirrhosis. So if it goes above 10 or 12%, you have a dramatic drop in the survival rate. Turns out that there's a link between oxidation of methionines with diabetes and diabetic renal failure. So we're seeing when you have an increase of exposure to air pollution, we have a dramatic increase in the oxidation of the cysteine 34, but also a large increase in a methionine oxidation. So it might be that, oops, wrong direction. It might be that this methionine and cysteine 34 are sensors of long-term exposure to oxidative stress. I'd also like to draw your attention to this novel adduct that was discovered on albumin. It's phycoerythrobilin, and we can prove that or validate that this adduct is there by comparing our discovery at an independent analysis of the adducted peptide with a more sensitive targeted analysis for that adducted peptide, and compare the intensities, the fragment ions and the relative intensities of these fragment ions are the same. And by looking at the mass, the high mass accuracy of these fragments to the predicted sequence, we're very confident that this adduct is actually present there. But why is phycoerythrobilin on albumin? This is a photosynthetic accessory pigment of cyanobacteria and red algae. And cyanobacteria is an indication of contaminated drinking water, whereas red algae is found in sushi. So the question is, this is something from dietary, or is there a drinking water contamination problem? So this is early times. We have several challenges left, many challenges left to really use this as an assessment of environmental exposure. One is validating that the adduct is what we actually think it is. Right now, we're basing it on the high mass accuracy of the mass shift and with known adducts in Unimod and in the literature. But really, we need to make standards to prove that they really are the adduct that we're detecting. Then we need to start correlating these adduct exposures with health outcomes. And then once you get a correlation with an exposure, you need to find out what the exposure is from. And then more importantly, once we know that an exposure is a health risk, it'd be interesting to follow up and look at what biological pathways are being influenced. Is this a cause or is this an effect of the exposure? So hopefully I've given you a glimpse that we have a novel way of looking unbiasedly at all the as many adducts as we can detect in albumin. We can both detect known and novel adducts. Not only detect them, that we can determine the relative abundance over time. And we're up to over 25 sites in albumin with over 120 adducts. Not only can we detect them and get the relative abundance, we can monitor the changes over time or with any kind of exposure. This can allow us to generate hypotheses to test and develop, hopefully, signatures of internal dose of external exposures. And what's also falls out of this is that although we're doing it on albumin, this approach is applicable to any protein that you can get in reasonable amounts to do a comprehensive look at what the modifications may be on. And we actually have started that with some other projects. So I'd like to thank my group at Hopkins, my collaborators in the public health and University of Michigan, Fred Hutch, and our funding, and you for your attention. Thank you. Yes.
So, just thinking, like future applications of this. So there's some places that where exposure to environmental toxins is high, in industrial areas and the population is exposed to those toxins. Would this be a way to kind of document that people actually have been exposed, in an environmentally risky area, or what do you envision?
Yes. Yes, exactly. Well, I mean, it's almost on the news constantly. This fire here, this train accident there, the train accident that happened in East Palestine. There is a assay that they can see whether or not they were exposed to the vinyl chloride, but it's a urine assay. So you have to take, you have to do it very soon after the exposure. But with this, there is an old report in the 70s that you can detect vinyl chloride on albumin. So we could have gone in there and said, these guys that live on this side of the street, yeah, you should be worried because you got exposed and we should do something about it. But these guys over here, you weren't exposed, don't worry about it. You can go on and be, you won't have a problem later on. So those kinds of things. The other way of using this is to inform environmental public health policy. So if we're detecting exposures in areas of high risk where there's a lot of industrial pollution, there may be ways to mitigate that either by health policy or by medical intervention. And then you could also track the efficacy of those interventions. That's our hope. Okay, thank you very much.